Good morning, colleagues. Is it
Okay.
okay? Okay.
Yes.
First of all, I would like thanks organizing committee for the opportunity to share our results with the international scientific community. And my talk will be about the usage of some computational biology approaches and chemoinformatic tools in the field of medicinal chemistry and in the field of evaluation of biological activities of some drug candidates. So it's known that, it's known that natural compounds is a valuable source of novel molecules for drug discovery. And one of the successful example of chemical derivatives of natural metabolites are CDO-methyl and its analogs developed by Professor Michael Sporn from Dartmouth, United States, which is now under the clinical investigations in patients with different disorders, mainly associated with the malignant transformation and inflammation. In Novosibirsk Institute of Organic Chemistry, our colleagues developed and synthesized structural analogs of CDO-methyl based on other compound, glyceric acid, the main component of licorice root. And our groups showed their high B activity in respect to tumor growth and inflammation, both in cellular and animal models. And considering that the majority of studies about the anti-tumor activity of CDO-methyl and its analogs, this works mainly concentrate on cervical carcinomas, and a little number of studies were about their effect on glioblastoma. And the aim of our study was to determine whether containing pentacyclic triterpenoids can block glioblastoma aggressiveness. And the main task of our work was, first of all, to synthesize the novel compounds, bearing, and to evaluate the permeability of novel compounds through blood-brain barrier, and to explore the cytotoxicity of novel compounds against glioblastoma, and to evaluate anti-glioblastoma potency of the compounds in non-toxic concentration. The distinct block of the study was focused on glial to mesenchymal transition, the main process associated with the diffusive growth of glioblastoma. And finally, we verified our results obtained in cellular models in animal model. Before the synthesis, we analyzed the literature and found that introduction of amide group at the position C29 of CDO-methyl significantly increased brain accumulation of the compounds. And based on this results, our colleagues, organic chemists, synthesized novel amides of siloxolone with different substituents. And firstly, we performed the screening of cytotoxicity of these compounds in different tumor cells and showed high activity. And after that, we concentrated our attention on anti-glioblastoma potency. And first of all, we tried to predict the capability of the novel compounds to pass through blood-brain barrier. And to address this issue, we used two chemoinformatic tools, ELF platform and pre-admit, and we showed that the majority of novel compounds can cross the blood-brain barrier, and moreover, these compounds are not the substrates of P-glycoprotein, the main efflux transporters in blood-brain barriers. And this data was further validated by us. So we selected compound 12, which was characterized by the highest BBB score in both models, in both tools, and showed that the injection of intraperitoneal injection of these compounds to mice lead to accumulation of the compounds in brain tissue, and we used for it HPLC MSMS analysis. So after that, We evaluated the toxicity of novel compounds against tumor cells of glial and neuronal origin and showed the high activity of these compounds. And we selected compounds 12 as hit compound and showed after that performed mechanistic studies which showed that compounds 12 induced death of glioblastoma cells by triggering apoptosis. So we showed externalization of phosphatidylserine after the treatment and activation of caspase 37. Along with the activation of apoptosis, we showed that hit compounds triggered autophagy in glioblastoma cells. Next, we checked the effect of triterpenoid on mitochondrial potential and showed that incubation of glioblastoma cells with compounds 12 significantly decreased the mitochondrial membrane potential, which was associated with activation of generation of reactive oxygen species. And further co-incubation of glioblastoma cells with hit compounds and N-acetylcysteine, it's a well-known antioxidant. In this experiment, we showed that the death of glioblastoma cells induced by hit compounds is ROS dependent. Based on this data and based on published results, we checked the ability of compound 12 to interact with active sites of mitochondrial protease along P1 because this protein is well-known protein target of cytidine-methyl, the structural analog of compound 12. And we show it by using molecular docking. We showed that hit compounds indeed can interact with the active sites with low binding energy and to form 3 hydrogen bonds with key amino acids of this protein. After verification of anti-glioblastoma potency of the compound in toxic concentration, we evaluated the effect of hit compounds at non-toxic concentrations in glioblastoma cells and showed that compound 12 effectively inhibits clonogenicity of glioblastoma cells and their motility. Moreover, we showed in vitro analysis that hit compound can effectively blocks the ability of glioblastoma cells to form tube-like structure. The process named vasculogenic mimicry is associated with neovascularization of glioblastoma. So our in vitro analysis showed multi-targeting complicated effect of our compound on different characteristics of glioblastoma cells. And after that, we verified this data in murine model. Murine model, xenographed model with U87 glioblastoma. And we showed that 7 intraperitoneal injection of compound 12 at dosage 20 milligram per kilogram effectively inhibits the growth of tumor, decreased the number of key 67 positive cells, and this dosage, this scheme was non-toxic for the animal, as you can see from this graphic. Yeah, and additionally, we verified that our compounds accumulated in brain of the mice. So we repeated experiments with HPLC MSMS. So and showed that yes, these compounds was accumulated in brain tumor of experimental mice. Okay. After that, we questioned which proteins can be considered as probable primary targets associated of compounds, hit compounds associated with its anti-glioblastoma effects. To address this issue, we used network pharmacology analysis. First of all, we predicted probable protein targets by using 2 chemoinformatic tools, Polypharmacology Browser 2 and Swiss Target Prediction. And after that, by using Cytoscape, we reconstruct gene association network from genes related to GBM pathogenesis based on DGNet databases. and predicted protein targets. And after analysis of created networks, we ranked targets in accordance with their centrality score. And top 4, the most associated proteins with targets with glioblastoma, you can see here. After that, by using molecular docking, we showed that EGFR, AKT1, and ARBB2 can be considered as the primary protein targets probable of hit compounds. And moreover, these protein, these receptors and kinase are highly associated with GBM pathogenesis according to Open Targets database. Okay, the next block of our study was about evaluation of the effect of the compounds, novel sulfonamides, on glial to mesenchymal transition, the process which is associated with a high aggressiveness of glioblastoma, because it's known that treatment of glioblastoma results in the change of morphology of glioblastoma cells to mesenchymal phenotype, which is characterized by high aggressiveness, high invasiveness, high motility, and resistance to radio and chemotherapy. And inhibition of glial mesenchymal transition can be considered as a promising, novel, promising approach in glioblastoma treatment. Let's return to our compounds. For this block, we selected amides which can penetrate the blood-brain barrier. Here you can see other IDs of these compounds. For example, GIL7, it's a compound 12, which I talked about previously. Okay. First of all, we identified non-toxic concentration of succinylamides in glioblastoma cells, and after that we evaluated their effects on several characteristics in glioblastoma cells activated by transforming growth factor beta. Transforming growth factor beta is well-known inducer of glial to mesenchymal transition, and we show that TGF-beta significantly decreased cell adhesion and increased the spreading tumor cells, the increasing of cell area of glioblastoma cells. And screening our compounds in these models revealed several amides which can restore these characteristics to control cells. For instance, based on these results, we selected hit compounds GIL46, for instance, this one and this one, which restored these characteristics to the control level. After that, we explored the effect of hit compound on the motility of TGF-beta1 stimulated glioblastoma cells and revealed high inhibition of the migration rate of tumor cells. And moreover, we showed the ability of compounds to inhibit expression of key mesenchymal markers of glioblastoma, for instance, encalcerin, fibronectin, and SLAC. And upregulation of OLIC2 is well-known glial marker in glioblastoma cells. And by using molecular docking, we showed that compounds GIL46 can interact with active sites of TGF-beta receptors type 1 and type 2 with a binding energy comparable with that of known inhibitors of these receptors. And after that, we evaluated the effects of hit compound on some characteristics associated with the diffusive growth of glioblastoma, notably the effect on clonogenicity and the spheroid growth of glioblastoma cells, and showed high inhibitory potency of the compounds on these processes. And finally, co-incubation of glioblastoma cells with our compounds and temozolomide, which is a first-line drug for glioblastoma treatment. In this experiment, we showed high synergistic effect of GIL46 with the temozolomide. So, taken together, our results showed that xalinon bearing triterpenoids can be considered as a novel platform to create novel drug candidates for the treatment of glioblastoma. And these compounds, the mechanism of these compounds includes different stages. So thank you for the attention, and I would like to thank my colleagues from our institute and Institute of Organic Chemistry and Russian Science Foundation for financial support of this study. Thank you.
Okay, thank you. Yes, there are a few questions. The question from Sofia Borisevich. Actually, I don't really understand. Ligand atoms interact
This question for Urashi Tivari.
Oh, for Urashi. Oh,
yes, we discussed it your talk in the chat.
Okay. Yeah. Yeah. I was a little bit surprised to this kind of question. Okay. So any other questions?
Novel message.
Oh.
Unfortunately, no. MD simulation. So we tried. Now we are learning how to perform it by using Ember or Gromax, and my students now are learning how to perform it. And unfortunately, no. But we are planning to perform this in the near future. Maybe a collaboration is a good idea. Oh, cool. So it will be good if we connect with each other to write, maybe to create a novel article together. Thank you.
Okay.
But it was answer on the question of Sophia to Tivari, not to you. Yeah. So okay.
So
maybe
it's a good way to find new contacts here.
Yeah, sure. That's one of the aims of the conferences, to find some new collaborators.
Yeah.
Yeah.
Yeah, I don't see more questions.
Yeah, so let's just wait a few 1 minute, before the second talk. Thank you once again, Andrey. Very impressive results.
Thanks a lot.
Good luck in further studies.
Yes. Thanks
Thank you very much.